Pat Stayton is the Washington Research Foundation Professor of Bioengineering. And in addition, in recent years, he's become the director of the new Molecular Engineering and Sciences Institute, a big effort at the University of Washington to drive right down the middle of this interdisciplinary area of engineering at the molecular scale. His research topics focus on applied bioengineering at the molecular level to both personalize and refine drug delivery. His partner in the lecture tonight is Professor Suzie Pun. Suzie is the Robert F. Rushmer Associate Professor in the Department of Bioengineering. And her research focus is on macromolecular drug and gene delivery systems. This promises to be very enlightening, and we're thrilled to have our two speakers with us. Please welcome Drs. Stayton and Pun.
Well, thank you, Lee, and good evening, everybody. It's exciting to see such a large room filled tonight to hear about such little things. Tonight, we want to go over one of our main visions for the biotech side of molecular engineering. This is our new building that we're so excited about, and believe me, it's also easy to get up in the morning and come into this new building. It would be great to have you there at one of our events if you're curious about seeing this. This building is one of the first buildings on campus that is really designed to bring teams of researchers together. It is not a department-based building. We have faculty from engineering, medicine, and arts and sciences all coming together around grand challenges. And the building itself is designed to achieve that in some new ways. I was very fortunate to help design the building with a great architectural group. And the labs, even when you come in and look at the labs are very different than most of the other buildings on campus. The labs are all very open. The people space is very open. They run parallel to each other, so it's easy to intermix. And in fact, we were able to design it so people have to collide. It's a collision building, I call it. And it happens with the coffee pot, where we put the coffee pot on upwards. The teams that are in there are the clean tech and biotech. The clean tech you've heard about, as I mentioned, and I want to give you an overview starting out, and then Suzie will come and tell you about some more exciting research in detail, but I want to give you an overview of one of the grand challenges in the bio area that we're very excited about. So the way that I've always looked at it, after kind of seeing how Lee did it so well for so many years, is putting puzzle pieces together. And again, what's so exciting about molecular engineering is that it's not based in any department, so we can kind of pull together the best people from across campus. I always say I've got the easiest job in the world. It's like take brilliant people, mix them together, and you get all the credit for all the good things that come out. But in this case, what I want to talk to you about is one of the grand challenges in the medical field, and that is to develop what we call biologic drugs. And I'm going to kind of go through the current setting that you face as a patient in the two major classes of drugs. But starting out, this is a big challenge, as I hope I'll be able to convince you of. And no one set of researchers from any one lab could ever hope to attack this grand challenge. We have people from bioengineering who make new drug delivery systems, and we'll give you some examples of those. One of the things that we were always missing, though, was pharmaceutical people. We have a great school of pharmacy, and yet we were never getting together with them. They kind of lived their life. We lived our lives, and we just had never really gotten together and say, what if we brought somebody who really knows how to develop drugs together with our kind of group that we think is good at making new drugs, but we don't know really how to develop them in a sophisticated way. So we're able to bring people from the School of Pharmacy, some really exciting faculty members in to build this puzzle. We also, of course, need the medical people, so we have clinical researchers involved in this team. And then we're really excited about the fourth puzzle piece, which is molecular imaging, and bringing that in. One of the biggest problems, again, when you work in isolation is you develop new drugs, you kind of put them in one of the model systems for a disease, and you kind of look for what you want to see. You look for success. And it may not really have the kind of success that would be meaningful to a real pharma company, but we know how to publish papers, and so you kind of get a paper out of it. But we don't oftentimes know what's not working. And molecular imaging has made some really tremendous advances that allow us to see where our drugs are going off target, so we can get a better feel for the failure mechanisms, what's going wrong, or for that little bit of it that's going right, maybe we could learn why it is. And so taking this all together, I'm going to kind of whiz through this and see how these different pieces are coming together in a way that hopefully would have this bigger impact here. We hope to develop new cancer drugs, new drugs for chronic disease. Diabetes is one, of course, for which there's a big pressing worldwide need. New drugs for brain disease, that's another huge area, and Suzie is one of the world's experts in gene delivery to the brain, and you'll be hearing a little more about that. But I think all of us are aware of the need for new drugs for things like Alzheimer's. And then lastly, new vaccines as well. Growing area, hot topic in Seattle, but again, one of the special new capabilities of molecular engineering. So what I want to do is first lay the groundwork with just a couple of slides to tell you how exciting the timing is. And then I want to go to kind of the challenge part. But from a timing standpoint, we're living in what you might call the genome era. The genome era has been it's kind of breathtaking, even for a researcher like Suzie and I who kind of work around the area. But sometimes I just go home and when I read, the paper, it's just astounding the breakthroughs that they're getting, and it's the understanding of biology in its complexity. And I stole this slide because it means nothing more to me than it probably means to you up there to look at this network. And this came out of a famous journal called Nature. And what they were studying is the things that start to go wrong in brain cells that give rise to brain tumors. And I think what I just have you appreciate is the level of detail. They're looking at all these networks that are switching on or off in the wrong way, and the end result is cancer. But the level of detail is astounding just from a fundamental standpoint. The problem, though, is that we have now lots of papers like these, but we still don't have any new therapies. And that is really our challenge that we're taking on. How do you convert this information into new therapies? And it turns out to be not so easy as people thought it might be. Another great example that I think is really transformative are new sequencing technologies. This is the sequence of DNA in what are called base pairs, GCTA. We now have many human genomes already sequenced. Even more than that, we have many individual tumors that have been sequenced. We have this incredible new data set of genetic information associated with cancer in particular, but many other diseases. We are beginning to get a real fundamental understanding of what is going wrong. That also gives you ideas about developing drugs that could be more specifically tailored to any given individual's disease. And I'm going to show you one such example today. But again, you hear a lot about personalized medicine, but we have very few examples. We have the information, but we can't translate it yet. And that, again, is what molecular engineering is really all about. We're the translational people who really want to get it out, to the patients. This is one of my favorite examples of a real big breakthrough that actually did lead to a new therapy. This is Kareem Abdul-Jabbar. For those of us who played basketball back in the this era. We always laugh at ourselves that we would ever wear shorts like that. But we did. That's an old picture, obviously. But Kareem Abdul-Jabbar came out just a couple years ago and announced that he had a type of leukemia. And with this particular type of leukemia, there's been some real genomics breakthroughs and an understanding of what goes wrong. Leukemia, the relevant cell is your white blood cells. And a very specific chromosomal rearrangement occurred in one of his white blood cells that is a precursor to produce all of his white blood cells. And it's a very specific DNA rearrangement in the chromosome. The details aren't so important. But the problem is that the new rearranged chromosome that they call the Philadelphia chromosome leads to highly dysfunctional white blood cells. And you can just kind of see that's not what a cell looks like, all those projections coming off. That's not a good thing, okay? The exciting thing was, though, that a group of researchers took this information and said, can't we use this understanding of that DNA change to come up with a new drug? And what happens in this kind of leukemia is that a rearrangement of the gene codes for an enzyme. And the enzyme, unfortunately, is always turned on, and it turns the cell on toward the tumor type. And so once this enzyme is created, we call it constitutively on. It just runs all the time and creates white blood cells that are cancerous. And they relatively quickly, come to dominate that part of your immune system. In the old days, there was very little you could do. You could do chemotherapy, but it was not a nice therapy, and it wasn't very effective at the end of the day. People came up with the idea that this new enzyme had a little pocket, and they designed a drug called Gleevec. And this was, one of the key developers was in Portland at the Portland Health Sciences Center. It's a very inspiring story about how they overcame a lot of odds and a lot of people not believing in them to develop a small drug that would bind into this pocket, and it turns off the enzyme. And so by attacking that genetic cause of that type of leukemia, there's been some really transformative effects on people like Kareem Abdul-Jabbar. He's been on this drug now for a couple of years and is doing very well. This is what we call a small molecule drug. This is what it looks like from a chemistry standpoint. But these small molecules are nice. You can formulate them into pill form so you can take them orally. Small molecules travel well through the body. They distribute throughout the body, and they also move into cells. It's also kind of their downside. That's why we have so many bad side effects with cancer drugs, is that they don't just have their effect on cancer cells. They also have some bad side effects in killing cells that we need, okay? But this was an example of a small molecule that bound very specifically to that enzyme that's only found in the cancer cells. So this has relatively good side effect profiles. And Time magazine even noted that this is, the first example of a new genetically targeted small molecule. And it seems like this ought to be the future, okay? About 60% of these types of leukemia patients have that mutation. And so about 60% of the leukemia patients can be tested for that mutation, and if they have the right type of genetic mutation, they're very nicely taken care of with this drug. And that's, a great story. This is what that molecule looks like. If you look at the structure in more detail of that target mutated enzyme, this is how it fits in there. There's a pocket, and we call it like a lock and key fit. And the better the fit, the more energy there is, and the better this drug is from a simple perspective. But this is wherein lies the limitations of this idea. It turns out that small molecules, for most of these types of targets, we can't design them to have enough binding energy because they're too small and their targets don't have this nice lock-like structure. So it turns out, unfortunately, that from first principles, while it's a great story, it's not going to get us where we need. One of the experts, Greg Verdine at Harvard, has estimated that as high as 80% of different disease targets can never be hit or drugged, we call it, with small molecules. So that's unfortunate. For the 20%, we want to get up there as high as we can, but that still leaves 80% of disease where this can't be our paradigm. Now, there's a second major class of drugs that are not small, they're large. And in fact, they're molecules that come out of our own biology. And they bring the power of biology, and they're called biologic drugs. And the main types that Suzie and I are going to talk about tonight are proteins, DNA, and RNA. Proteins have already had a big impact. A decade ago, there were none in the top 10 best-selling drugs. I just checked a couple weeks ago, and 7 of the top 10 best-selling drugs are now proteins. So they're having a great effect, actually. Biologics are here. You're living in an era. My dad is on one of these protein drugs from Amgen for prostate cancer. They have a real impact. Probably many of you know people who are treated in the cancer field with some of these drugs, or the rheumatoid arthritis field, or diabetes with insulin. Those are protein drugs. Now, the other major category are DNA and RNA. And I just want to give a very quick primer. This is called the central dogma of biology. If we look in the nucleus of cells, the genetic information is stored in DNA. And DNA gets read out into proteins that do most of the work in biology through a secondary molecule called RNA. So DNA codes for RNA, codes for proteins. If we think about these three, these are our three kind of biologic candidates for drugs, and we're going to be giving you some examples of all three. DNA is maybe the more famous example in gene therapy, and Suzie again will be talking to you about it. Gene therapy, one of the exciting ideas of gene therapy is that they can take defective genes, and this is one of the most famous cases I'm sure you've heard of, the bubble boy syndrome, where these poor little boys are born with a genetic mutation in a key protein. So the idea with the gene is if we can replace a correct form of the gene, then our body will read it out into the correct form of the protein to correct that. And they've made some very exciting progress. They've done gene therapy on their stem cells, and they put these back in, and these genetically modified cells will produce the right form of this enzyme, and it is having a favorable impact. And it's just one example. Suzie will give you another nice example. But that's how the power of genes work. Very different than small molecule drugs. RNA it's hard to find in the biotech world right now, a more exciting example, more interest. RNA has the same general idea. Again, just a reminder, the DNA is read out into RNA and it codes for proteins. But there's a new type of RNA that was discovered, a new pathway for regulating this process of going from DNA to proteins. What it basically does is cleave the messenger RNA, very specifically. And what this allows you to do is at the RNA level, knock out the activity of particular proteins. And so you can knock out defective genes, knock out defective proteins. And for example, rather than giving that small molecule drug that blocks Kareem Abdul-Jabbar's enzyme, you could just knock out the RNA and knock out the protein so it would never be made in the first place. Okay? That would be the promise of a biologic drug compared to a small molecule drug. So taken in total, the exciting thing about biologic drugs is they have way more upside and way more potential than small molecules. RNA drugs can work against any disease in principle. They can knock out any target in principle. Monoclonal antibodies, proteins, can bind to most any target and neutralize it. DNA constructs can express almost any target gene. And so they have a dramatically increased specificity and target range. They could get us to that missing 80%. Okay? So that all sounds great. And it is great in principle, and it's why we have come together at the Molecular Engineering Institute to try to focus on developing biologic drugs, because it's so clear that's where the future ought to be. But now let me tell you the problem with biologic drugs. You're probably waiting for the punchline, right? There are some problems, and they're big problems. Currently, all protein drugs work on targets that are on the very outside of cells. They simply cannot travel to the inside of cells where about 90% of target space lies. So we are already encountering, while there's been a huge growth of protein drugs, we're already beginning to see limitations. They don't travel well. They're like my dad. They don't travel well. You know, they just, small molecules go everywhere because they're small. Proteins, DNA, RNA don't travel well. Another classic example, I get you excited about RNA. There are no RNA drugs right now. RNA is very difficult to get to the cell, and then their target is inside of cells. One of the Nobel laureates who discovered this form of drugs, Phil Sharp, says it very simply. The issue is delivery, delivery, delivery. The first gene delivery drug was just approved. One, for all that you hear about, gene therapy. One, and it was just approved this year. And it's not anything that's going to change all of disease space, okay? It has to get inside of target cells. We have no oral biologic drugs. We can't put them in pills. They're very different. If you put them in the gut degrades them. Small molecules escape, go where they need to go, not biologic drugs. And lastly, I wanted to highlight this because of the exciting work Susie's going to tell you. There are no biologic drugs against the brain. The reason is there's something called the blood-brain barrier. We cannot get past it. Nobody knows how to get the biologic drugs. There's some very exciting local delivery examples for brain tumors. If we just directly inject the antibodies, you can show nice effects. But that is very limiting. We need to be able to deliver them through more normal routes and get them to the brain. So this is what I call the conundrum of biologic drug potential. This is a plot of the percent of drug sales, all drugs. This goes across all diseases. The blue bars are small molecules. You can see which is the dominant current drug. It's small molecules. These three colors you can kind of look together are biologic drugs, proteins and vaccines. Now you can see growth. They're becoming more important. And like I said, 7 of the top 10 bestsellers are proteins. That's because they cost $100,000 a patient. They're pricey. My dad just had this done. They're pricey, but they work. They're very nice drugs. So a question is, can we sustain this growth, and what's the upper limit? A lot of people are very bullish on this, but they're not really thinking about it. The truth of the matter is that these are going to plateau unless we can deliver proteins to intracellular targets, and unless we can broadly open up DNA, RNA, and RNA drugs. And so this sounds a little dramatic, but it's been funny. Susie and I have been through a recent large effort to get a big national-funded biologic delivery center. And it's become clear to me in a way that it wasn't so crystal clear to me. But going through this process, this is just the truth of the matter. Drug delivery systems are necessary to open up biologic drugs to intracellular target space. So if we do not succeed at developing drug delivery potential, then the genome era will be a set of interesting newspaper articles and textbook chapters on how smart we are in understanding biology, but it will not be translatable to patients. Only drug delivery solutions can fully unlock the potential that's there. So this is one of the great grand challenges of our time, and I don't mean it to say we're the only ones doing this. It's a worldwide effort to do this. It's also critical to our country. We have had an incredible leadership privileged leadership position in this country that is our pharmaceutical, biopharma, biotech industry. We generate the IP related to more than half of new medicines in the US. We account for 80% of the world's R&D in biotechnology. We're a rare source of growth in manufacturing jobs. Seattle actually has some really interesting companies that manufacture these, they're jobs for our students. It's a big driver of both our healthcare, but also it's an economic engine. I think you can guess where the challenge is coming, though. It's an unprecedented time around the world. We're not the only ones who recognize the potential, but the challenges. And the U.S. does not have all the answers in this field. If you look, this is a title of an article from China, 5-year biotech investment fires clear warning. 5-year investment started last year, $310 billion US, with the goal of making China the world's pioneer in biotechnology. Korea made biotech their top national area of investment over IT as of last year. And they're good. Many of their students are educated here and are going back to Korea and China. Okay? India, Brazil. It's a new world. It's not simply us versus them. There's also this unprecedented opportunity there. Our companies are there. Pfizer, Merck, large, major investments in China, in Korea, in India. So I wanted to mention that this is another thing that really interests me, and I think we could have a lot of impact here. We're going to create global education opportunities because that's going to be the playing field, for my son, who's going to be a scientist, and he's going to work on these problems someday. He's 9, he kind of knows it, but he doesn't quite know how certain it's going to be. They're going to have this global playing field, and we have to give them the setting. And so one of the exciting things about the building is that we're bringing in a lot of international collaborators so that they learn to work across cultures. And believe me, I get the email notes. There's some issues that come up. And it's very important that they learn to work in this setting and they can see the opportunity and the competition. It goes together. So I'm going to move real quickly through these slides. People ask me, this is a question my mom goes, well, why is it so hard, Pat? Why is it so hard for proteins? Well, it's because, if you think about it, these biologics are injected into a whole person, and you got to think about your systemic goes throughout your whole body. At some point, it gets out of the circulation, and say we're looking at the brain, it's got to move into the brain. There's another set of systems and barriers at that level, but eventually it has to get all the way down to the cell level. If you do the math, that is an incredible journey that's the equivalent of one of our students going from the Earth to the moon in distance. That's how far it is relative to their size. But it's even harder because there are cells, there are other proteins that want to get rid of that rocket before it reaches the moon. It's just incredibly difficult biologically to make this happen. There's only one success story that we know of, and it's not us yet. It's pathogens. Pathogens have highly evolved drug delivery systems, and the systems is really important. They have targeting. They have elements that get them through this level. They have elements that get them through tissue, and they have elements that get them to the right place inside the cell. We could only dream about it. They dynamically are engineered, they're engineered to dynamically interact with biology. They use our immune system. We know it can be done. It tells us we could do it. So we have to build these kind of systems. This is one quick example from my lab where we got excited about looking at the flu virus. This is an example of the system. If you think about it, their drug is RNA. But they have a carrier system. They have some parts that are to get inside of cells. They have other parts that are to get them to the right cell to go to the right place. And so we're building mimetic delivery systems by engineering polymers that have these kind of activities. It gets them inside of cells in a new way. It carries our drugs. And it has targeting so that we can focus on the cancer cell or other types of cells. One of our biggest interests is in this new class of RNA drugs. We know it can attack diseases in a new way. They can be tailored to individual genetic changes. It's the beauty of it. And it's very short in development time. Once we knew what a mutation was in a target, in 2 weeks, you can develop an RNA drug against that new target. The problem is delivery. This is one of the discoverers and Nobel Prize winners for this type of drug. Delivery is the major issue in it all. We've been interested in ovarian cancer and using this as a new approach. Ovarian cancer remains the most lethal gynecologic cancer. Median 5-year survival rate is 30%. Presents with local spread within the abdominal cavity. It goes from the ovary outward into the abdominal cavity. It's currently treated by injecting small molecule drugs into that cavity, and you can see from this number, they're just not effective. One of the key receptors that some of you have probably heard of is the HER2, called HER2, and it is the one that we've chosen to develop this carrier system toward, to deliver this RNA inside of ovarian cancer cells. But it's also, I put the breast cancer logo up here because this is the common marker found in breast cancer, and that's another beautiful thing about these kind of drugs. If you're successful in one area, it seems like it ought to be translatable to, say, breast cancer as well. What we do in the animal model is take human ovarian carcinoma cells. They express this receptor called the HER2 receptor. We have an antibody that has actually been commercialized already. Some of you might have heard of it, Herceptin. It's sold by Genentech and Roche. And we put that onto this delivery system with the drug. And the hope is that this will be targeted to the ovarian cancer, get inside, and knock down a key gene that is associated with cancer. These are injected into the abdominal cavity, and so they closely mimic the nature of the human recurrent ovarian cancer spread in that cavity. Here we put a light-emitting ovarian tumor in 3 mice just so you can see when you inject them, the tumors actually are distributed all over the abdominal cavity, and that again is the problem in real-life ovarian cancer. And then what we do is put an antibody that recognizes these tumors. We put an RNA drug. And the first point here is with the carrier, after 24 hours, if we red label our RNA drug, you can see that the drug is with the cancer cells. If we just have salt solution or the RNA drug alone, it's gone. It's immediately gone. And that, again, is what's so important about this carrier system. It gets it to the cells and delivers the drug to those cells. And if you look at the expression of a gene inside of those cells, this is ratioed to 1 here. You can see with an RNA that's very similar but doesn't match the sequence of the target, you have no effect on that gene level. But with the carrier system, we get greater than 60% knockdown in that ovarian cancer model. And again, it's because of the activity of the carrier of getting it to the right cell and inside the cell to its target. So that's our idea. It's going to take a lot of different kinds of people to put this puzzle together. And we're really just, we've kind of all been working in our little areas, you know, and doing things. But the hope at molecular engineering is to bring a much bigger power to this and to bring all the different sides of it, the pharma people, the clinical people, the imaging people, and those of us who actually make these carriers that can mimic the functionality of the pathogen systems. Lastly, I wanted to note that a big part of our institute is really meant to get it out of the academic setting. We have a big emphasis on interacting with biotech and biopharma, on startups, on licensing, and that's our mindset, is to come together, develop it, and get it out to people who can develop it and get it to patients. And this is part disclosure, but also an example. We have a startup company in Seattle called PhaseRx that is trying to industrialize and commercialize those carrier systems for RNA into cancer therapeutics now. So that is an introduction, a whirlwind introduction. What we're going to do now is have Suzie come up and give you a little more specific examples on the gene delivery side, especially. And Suzie is one of the world's leaders in the development of these kind of nucleic acid drugs and has a special interest as well, as you'll see, in brain delivery, which has got to be one of our most important challenges now, of course. And so I'm happy to introduce my colleague, Suzie Pun.
All right, so Pat gave you one beautiful example of an engineered delivery system for cancer. And I'm going to start with another example that would work hand in hand with the types of materials that Pat is delivering. So here is a schematic of a tumor, and the yellow cells are the tumor cells. So these are the cancer cells, and these are the cells that all of the current chemotherapies target. These cells are, again, responsive to chemotherapy, but one of the difficulties of targeting these cells is that they're genetically unstable, which means that they change over time. That's why, if you're diagnosed with cancer, the scariest part is when it reoccurs, because it gets much more difficult to treat once the cancer comes back, because the cancer is tougher and more difficult to kill. Now, if you look around here, what you see is that the tumor cells are not isolated, and there's a bunch of other types of cells that are in that environment. And the cells I want to talk to you about today are these cells that are shown in green, and these are the tumor-associated macrophage cells. Now, macrophages are part of our immune system. They're a type of our white blood cells, so they're our normal cells. But when these macrophages come into the tumor, they actually come and they help support tumor growth. So there's been studies that have shown that macrophages promote tumor growth. They promote metastases, which is when the tumor travels and can lodge someplace else. The macrophages help the tumors evade the immune system. So again, when we have tumors, these are uncontrolled growth in our body. And the question is, why does our immune system not recognize this and eliminate it by itself? And the macrophage is one of the components that helps to hide the tumor away from our immune system. And then finally, there have been studies that have shown that the macrophages actually help in chemotherapy resistance of tumors. So what we'd like to do is to target therapies also against these macrophage cells. And what we hope is that if we can kill off the macrophage, this might be a wonderful adjunct therapy to some of the chemotherapies or the cancer cell targeting therapies that are being developed. So how can we do this? Let me tell you a little bit about macrophages so you can see the dilemma that we face. So macrophages come from blood-derived cells. They come from these cells that are in our blood called monocytes. These monocytes go into the tissues and they become macrophages. And what you'll see is that these macrophages are present in all of our resident tissues. And they're known by all these different types of names. And you might have heard of some of these names. But these are very important cells. They reside in our tissues, and they help to prevent infection, and they help to protect our tissues. Now, sometimes if there's inflammation or if there's an infection, our tissues will send out signals that will recruit new macrophages. And the type of macrophages that get recruited depend on the type of signals that are being sent. So there are two broad types of macrophages. One that is called the M1 macrophage. These are very inflammatory macrophages. And the second type is called M2 macrophages, and these are called anti-inflammatory macrophages. They help the body to settle down, and again, to settle the immune system. So the tumor-associated macrophages that I talked to you about earlier are part of these M2 category of macrophages. So you can see our challenge here. What we want to do is to target this very small subset of macrophages, but not hurt these other macrophages that are very important and that reside in our tissues. So how can we do that? How can we engineer a system that will very specifically destroy a small population of macrophages? So what we do is we can actually generate these types of cells inside the laboratory, and we have done this. We take monocytes from the blood, and we give them these signals that cause them to become M1 and M2 macrophages. And then what we do, I wanted to point out that these are actually important cells in other types of chronic diseases. So I mentioned to you that these M2 cells play a major role in cancer. They also have been implicated in asthma and lung fibrosis. And these other types of cells, these ones that are chronically inflammatory, have been implicated in diseases such as obesity and also atherosclerosis. So these are very important cells. They play an increasingly appreciated role in various diseases. So again, how can we find targeting elements to this small population, this subpopulation of macrophages? Well, we turn to a molecular biology technique known as phage display. These take advantage of viruses that infect bacteria. And these can actually be commercially available. These are commercially available. These are libraries of phages that have over 1 billion different peptide sequences. So you can buy a library. It has a billion different motifs. And we use this to screen for the motifs that have the desired properties that we want. And the trick is that the motif is contained within the DNA, so then we can go back and figure out exactly what the identity of that motif is. So here's an example experiment that the students in my lab perform. So we take this enormous library, 1 billion different sequences. We first take this library and we expose it to the cells that we want, the normal cells. So these are, for example, macrophages that we don't want to target. And we take only the phage that don't bind to those cells. And then we take that remaining library and put it with our M2 cells, the cells that we want to target. And from that population, we take out the very few phage that bind very specifically. And we do this over and over and over until we are able to find a sequence that's very specific. And so what I want to show you here is that we have successfully identified a peptide that binds very specifically to these anti-inflammatory macrophage. And it's so specific that you can see it doesn't bind to these other types of white blood cells that you would think might be very similar. So these are another type of macrophage, and these are other white blood cells that are present in the body. And so we show that we can use this macrophage now, we can use this peptide now to deliver specifically to these types of anti-inflammatory macrophage. So these are microscopy images of the cells, which are shown, the nuclei are shown in blue, and they're outlined here with a macrophage marker in red. And the protein that we're delivering is shown in green. And you can see here very clearly that only the peptide that we found, and not a scrambled control peptide, will enter these macrophage cells, and it leaves alone these other types of macrophage cells that are the pro-inflammatory type. So that's the first element that we need. So first, we need something that will target those cells specifically, and secondly, we need an entity that will kill those cells. So for that, we've turned to the cytotoxic peptides that were found initially to kill bacteria. These peptides are not able to enter the cell, but if you can get them inside the cell, they're very, very toxic. All right, so this goes back to the question that Pat was talking about. How can you take a peptide and deliver it into the cell where it can have its desired therapeutic effect? So together with Pat's lab, we've developed a type of chemistry that allows us to string together in a single material a bunch of different peptides. And so we have been able to do this now to string together these targeting peptides and then also these killing peptides together into this one drug carrier. And I want to show you some of our in vitro results. So this is a typical cell killing curve. So here we have the peptide concentration and then cell viability. And so what you want to see is that the peptide at certain concentrations will kill off the cells. The lower the concentration, the more potent the drug, right? It requires less concentration of the drug to kill the cells. So the toxic peptide alone we see is not very potent. It takes a very high concentration to kill kill the cell. Once we add in the targeting ligand, we can move that down a little bit because now the peptide is able to get inside the cell and it's able to kill the cells. And now look what happens when we put it together with our polymer material. This polymer is special because it can carry a bunch of these peptides together, and it can also carry a bunch of these targeting ligands together, and that makes these drugs a lot more potent. And you can see that in 2 different types of polymers that we've synthesized, we've been able to move down the concentrations by about 20-fold. So now that we have this targeting construct, we're testing these in these cultured cells, as I showed you in that data, and we're also testing them in animal models to see if we can potentiate traditional chemotherapy drugs. The second example I'll give you is for nucleic acid delivery. And Pat gave a great introduction to this. Our goal here is to be able to deliver nucleic acids into the brain. And we're interested in this because there are a lot of diseases that affect the brain. And this is only one subset of diseases, which are the neurodegenerative diseases. Okay, so you may be familiar with some of these: Alzheimer's, Parkinson's, Huntington's, ALS, which is also known as Lou Gehrig's disease, and spinal muscular atrophy. Now, there is a tremendous cost of care for these diseases. You can see here it's over $1 billion per year just in the United States to care for the individuals that are afflicted with this disease. And this cost of care is so high because there's very limited treatment options. And so these are really palliative, trying to make quality of life better for these patients. But to date, there's really no cure for any of these diseases. Now, as Pat said, the genome area has brought really good news because there are a lot of the mechanisms or the causes of these diseases are now being understood. And the question is, how can we translate that into therapies? So I want to give you one example of a successful gene therapy trial. This is a trial initiated by a professor at University of Pennsylvania. And in this case, she was taking gene therapy to treat congenital blindness. So these are children who lack a specific protein in their eye, and it results in increasing blindness over time. So what they've done is they've been able to put this gene into a virus and deliver it by direct injection into the eye. And they first showed that this was very successful in blind dogs, and they recently moved into clinical trials. And so I wanted to show you some of the videos from this. So this is a child who was first treated in just one eye, just to be safe. So they first started with treatment in just one eye, and 90 days later, they came and they brought him in to go through a maze. And they would patch one eye and then patch the other eye. And so this first example, they're patching his treated eye so that he's looking through his untreated eye. And he's trying to go through this obstacle course. And the goal here is for him to be able to follow these arrows and then exit. If I could turn the sound down on the computer, please. So you can see that this boy has his eye patched, and he's having a very, very difficult time going through the maze. And he becomes very, very frustrated through the course of this experiment. And you'll see him saying, this is hard, this is very hard, and he gets stuck. And he can't move and he can't progress. And it's extremely frustrating. I can't see anything, and he can feel he's running into things, but he's not able to progress through the maze. Now the second video is taken where they've now patched the other eye, and now he's looking through his treated eye. And you can see the maze a little bit better here. And you can see just how amazing his progress has been from that treatment. So he's able to follow the arrows, and then here's the door at the end. And you can feel the excitement from the investigators in the room at that tremendous progress in the child. So I wanted to just give you this as an example. Pat talked a lot about the potential of these type of therapies, and I hope this gives you a visual of just a remarkable progress in a disease that previously was not treatable. Now, the eye is a very special place. You can do a direct injection into the eye, and it's a relatively protected place. And the question is, how can we take this type of approach and extend it and broaden it so that it can treat a host of diseases? And again, we're interested in trying to translate this into the brain. Now, this therapy used a virus, but there's a lot of downsides of using viruses in other parts of the body. And I won't go into the details, but our lab is trying to develop synthetic materials that would mimic the action of these viruses, because we believe these types of materials will be safer and they'll be much easier to translate clinically. But again, we're faced with the same problems. These materials have to take the nucleic acid and navigate it from its point of injection all the way inside the cell. Not even just inside the cell, but it has to go all the way to the nucleus of the cell. So what we want to do is to apply chemistry techniques and engineering design techniques to make a material that can accomplish all of these steps. So I want to give you one example here of a material that we've made in our lab. This is drawn as a schematic because I just want to highlight the functionalities of this material. So first, we have a material that has a domain here in red that will bind and protect the nucleic acid. And that's shown here in the final form. Once we put it with the nucleic acid is condensed and packaged in the center. We need to have this blue domain here. This helps to protect the material as it's circulating in the body. And this is actually a catch-22 because this material is necessary to protect the drug, but this material also prevents the drug from being accessible inside the cell, right? So we then designed a linker here that is specifically cleaved inside the cell so that this protective shield is then uncloaked, right? So we have this particle that's protected. When it gets inside this cell, it takes off this shield, and then it's available to deliver the nucleic acid. And then finally, on the other end, we've put on here a little entity that allows it to recognize our cells of interest. In this case, we want to get it into neurons. And so this is a small peptide that binds quite specifically to neurons. And so to put it together, this is what we hope, is that we have neuron cells, we have our delivery vehicles. The targeting ligand will allow it to attach quite specifically to these cells. These particles will get taken up inside the cell. And after they're taken inside the cell, the linker will be cleaved, leading to uncloaking and then delivery of the active drug. So I want to show you that we've actually delivered these to the brains of mice now. And you can see that by combining these rational engineering approaches, we've been able to synthesize materials that are quite effective at brain delivery, or at least more effective than the baseline. So here we have the initial material where the cloak stays on and it's not able to effectively release the drug, and it's also not targeted. The green here shows the targeting, and so these are stably cloaked materials, and these are materials that can uncloak. And so what you can see here is that if you compare here between untargeted and targeted materials, you see that the targeting increases delivery efficiency by about 2-fold. And that if you have materials that can uncloak and take off that shield, we increase delivery efficiencies by about 6 to 8-fold. And if we put that together, that winds up being about a 20-fold increase in delivery efficiencies. And again, this has come about through progressive and iterative design, but understanding the biological system, partnering that with chemistry techniques, and then being able to test these in our animal models. So I wanted to summarize here with a couple of take-home messages for you. First, I hope, and I know that Pat has shown you the enormous potential of biologics and their ability to open up a wide range of new therapeutic targets. I also hope that he's shown you how critical engineered delivery systems are in order for us to realize the potential of these types of therapeutics. And also that the University of Washington is uniquely positioned to take a leadership role in developing these types of delivery systems. So you can see already from Pat, he's been leading us. I'm just one investigator in this group of people that he's put together to solve this grand challenge. So with that, I'll stop and ask for Pat to come up and take some questions from the audience.